we done this session about cytokines and MHC molecules, antigen presentation, and now in the third session we want to talk about innate immune system, innate immunity, and inflammation. Our goal of this class is to know what is the innate immunity and its features, and also we want to be familiar with some innate immune cells and also innate immune molecules, their roles in our body, and finally we'll talk about inflammatory responses, its steps in our body, and the importance of inflammation in our body. And you know, I told you we have two branches in our immune system. Okay? Innate immune system and adaptive immune system or acquired immune system. You know, when some pathogen enters to our body, innate protection against pathogen is made by sequential and coordinated responses of innate immune system and adaptive immune system to protect us against pathogen. And what about the features, general features of innate immune system? You know, innate immune system as a first line of defense against pathogen is also very called as natural immune system or native immune system or immunity. Why we say innate immune system?
Because it's already there.
Yes, exactly, it's correct. Because is mediated, is defense against pathogen through different mechanisms are present even before pathogen enter to our body. And also, innate immune responses start a few hours or days after entering pathogen. So it's really fast rather than adaptive immune responses. And finally, we can say innate immune responses are non-specific rather than adaptive immune response. You know, adaptive immune response because of presence lymphocyte B, lymphocyte T, and a lot of having a lot of TCR and BCR on their surface, and they can specifically recognize the pathogens, peptides enter to our body. But in the innate immune system, immune cells with some mechanism are present before entering pathogen to our body, are able to non-specifically recognize the pathogen, and so we can say innate immune system is faster than adaptive immune system, non-specific against different pathogens, and we can say fights all pathogens at the same way, and also is called natural and native, the other names of innate immunity. Take the right picture, for example. In this slide, I want to explain about collaboration between innate immune system and adaptive immune system. In every place you are living there, for example, in Kish, when some criminals enter here, so the first line of defense is police. Police try to catch the criminals, and without paying attention to their crimes, catch them and imprison them, and after that introduce them to specific sites like courts. in every country, in every city. So, like this story I told you, in our body, when some pathogen enters our body, the first police in our body are innate immune system, and this innate immune system infers on these foreign characters, these foreign pathogens and substance, and so after that they hold adaptive immune cells, B cells, T cells, and aware them of presence some foreign substance and pathogen in our body, and after that these lymphocyte B cells and T cells come there, and they recognize every criminals, every pathogen specifically, and kill them or protect us against them. And in the left picture, you are seeing the collaboration between innate and adaptive immune system in reality. And as you are seeing, when some pathogen enters to our body, innate immune system start producing a lot of molecules and start recruitment of a lot of innate immune cells like neutrophil, macrophage, and through different mechanisms try to protect us against pathogen and control the spreading of pathogen to the other side of our body. And simultaneously, innate immune system called adaptive immune system, and through producing some cytokines I mentioned in the first session, alarm adaptive immune system and introduce these pathogens to adaptive immune system. This system is also activated and start producing some antibodies, effector T cells, cytolytic T cells, and through them try to defend us against different pathogens, deal with different pathogens. And fascinatingly, this adaptive immune system also produce some cytokine, as you said, as I said, you produce a lot of cytokine, and through producing these cytokines affect innate immune system. For example, I told you lymphocyte T cell produce interferon gamma, and this IFN gamma affect macrophage and enhance phagocytosis of macrophage cells. And what about innate immune system? You know, innate immune system defends us against, protect us against pathogen through three line of defense. The first line of defense is some barriers in our body, chemical barriers, physical barriers. Yes, skin is kind of physical barriers and biological barriers. And in the second line of defense, when the pathogens we can break these barriers, and passing these barriers can exposure to some immune cells, innate immune cells like NK cells, macrophage, monocytes, and mast cells, dendritic cells. So this second line of defense is cellular component of innate immune system. And in the third line, we have a lot of soluble proteins like complement, antimicrobial peptides, and some opsonins. All of these three lines of defense as a component of innate immune system collaborate with each other to protect us against pathogen and to control spreading the pathogen to the other side of our body and introduce them to adaptive immune system. And let's back to physical barriers. First line of defense in innate immune system. As you are seeing, skin, nasal hairs, and mucosal membrane in reproductive system, in respiratory tract, in urinary tract, and also in gastrointestinal tract. All of them are type of physical barriers. For example, In the skin, we have three layers. Every skin has three layers, like epiderm, hypoderm, and derm. And in our skin, there are a lot of, in epiderm, there are a lot of keratinocytes, and these keratinocytes are aligned with some dead skin cells. And these dead skin cells frequently are shedding and replacing. And so, because these keratinocytes have a tight junction with each other, so they are highly resistant to water and also any growth of microbes in their surface, entering microbes from their surface. And so these skin as a first physical barrier in ours, one of the physical barrier in our body, protect us against entering pathogens to our body. For example, a person who burn his skin, so this person is prone to different infections because this person missed his physical barrier, one of his physical barrier, his skin. So skin is very, very important for protection us. And another nasal air is beneficial for us, and because filter all of the dust, dirt, and microbes, pathogens in the air, and protect us to enter against these pathogens and block entering them to our respiratory system. And what about mucosal membrane? You know, mucosal membrane consists of many epithelial cells. These cells are able to produce some substance like mucus. Mucus is a slimy substance and can trap the pathogens inside itself, and so direct the pathogens out of our body. For example, in our respiratory tract, your septum is a kind of mucus along with microbes, and this combination, this substance direct the pathogens that have been entered to our respiratory tract out of the body. And but about the other barriers in our body is chemical barriers as the first line of defense in innate system. You know, in some surface in our body, in some fluid in our body, we have some chemicals that these chemicals have antimicrobial activity. For example, in our sweat, tears, and mucous, skin, and soluble, we have some enzyme. It's called lysozyme, and this lysozyme has antimicrobial peptide activity against different pathogens, and is able to destroy the cell wall of pathogens. And also the other chemicals are present in this area are cathelicidin, defensin, and bacteriocin, histatin, and the other type of chemicals. And another area in our body is serum or ear wax, and these wax also contain some antimicrobial peptide, and also because of low pH in this area protect us against pathogens, because inhibit growth of pathogen in here. The pH is between 3 and 5. And gastric juice is another type of chemical barriers. You know, gastric so much low pH in our body, about 2 to 3 is the number of pH in gastric juice, and if these gastric juice pour on your skin, so you experience skin burning because it's a really low pH, and this low pH can destroy every pathogen enter to our body. And another barrier is urine is acidic, and because of low pH can kill all the pathogen. And the other fluid in our body is serum. Serum is saturated with a lot of nutrients, and interestingly, also these nutrients are necessary for our body and are used by many pathogens. But normally in our serum there are unsaturated fatty acids, that these unsaturated fatty acids are able to inhibit growth of microbes in our serum. And another barrier, apart from physical and chemical barriers, in biological barriers in our on our skin surface, and also in our gastrointestinal lumen, we have so many bacteria, beneficial bacteria. They aren't pathogenic, they are beneficial bacteria. They are called normal flora or microbiota, and microbiota is very, very important field of research these days, because all of the scientists in different major are reporting the association with microbiota with specific diseases. For example, association between microbiota with cancer, association between microbiota with autoimmune disease, and these microbes, apart from the other roles in our body, such as regulation and modulating of our immune system, can be first line of defense against pathogen, because they compete with pathogenic microbes to nutrients and sites, and so because they overcome these pathogens, and so they inhibit growth of these pathogens in our gastrointestinal lumen, on our skin. And but sometimes when you eat antibiotics a lot, more and more, without any prescription, after a while you may experience some infection, and you will be disposable against different pathogens. Because you missed your microbiota, and this microbiota normal flora is very important. And also, apart from competing for nutrients and sites, these normal flora or microbiota are able to produce some antimicrobial peptides like bacteriocins, and these components, these chemicals are able to kill the pathogens and have antimicrobial activity. And but about the second line of defense against pathogen in innate immune system, I mentioned is different immune cells like phagocytic cells, macrophage, neutrophil, dendritic cells, NK, eosinophils, mast cells, and innate lymphoid cells. Innate lymphoid cells are like lymphocyte T cells, and we have different subtypes of innate lymphoid cells in our body, and they are classified as innate immune system, but they helper 1, T helper 2, T helper 17, and protect us against pathogen as first line of defense. And what about PRR, pump recognition receptor? In previous session, you asked me about recognizing antigen by immune cells, how these immune cells are able to recognize different antigens and how immune cells respond to different cytokines. You know, in our body, innate immune cells, like adaptive immune cells, have many receptors on their surface. These receptors are called hand recognition receptors. And these receptors can be expressed on the surface, on the cell membrane surface, can be in cytosol, or can be soluble in blood circulation. So we have a different type of hand recognition receptor, or PRR, as a receptors of innate immune cells in different locations. And but what is the ligand for these receptors? PRR. Okay, in short, I say PRR. What is the ligand? The one type of ligand for these receptors are PAM molecule. PAM means pathogen-associated molecular pattern. Okay, and another type of ligand for these receptors are DAM, image-associated molecular pattern. DAM are some antigens, some molecular structures on the surface of apoptotic cells, imaged cells, aged cells, and sometimes some self-antigens in our body, some self-proteins or ions in our body are produced in huge amounts. So these are called DAMs. Okay. For example, ATP can be DAM. Okay, and NaCl can be DAM. Whenever these structures are produced in huge amount in our body. So these PAM and DAM, after binding to these PAM recognition receptors as soluble molecule in blood circulation, as a cell membrane receptor on the surface of, in the cytosol of cells, after binding to its receptor, lead to some signaling, starting initiation of signaling pathways in cells, and lead to killing of microbes through phagocytosis, through producing cytokines and releasing cytokines, through degranulation of cells, and finally lead to inflammation, because some of these receptors lead to activation of innate immune cells, and these innate immune cells, after receiving activating signals, start producing chemokines, and these chemokines lead to recruitment of immune cells, so lead to inflammation. And I told you we have different types of PRR. Some of them are TLR, or toll-like receptor. Some of them are CLR, or C-type lectin receptor. Some of them are scavenger receptor. Some of them are cytosolic in terms of their location, and they are called node-like receptor, or NNR, and RNR, or rig-like receptor. Some of them are soluble molecules, like pentraxin, like colectin, like fibrillin. So each of them have different different subtypes. For example, we have 10 subtypes for TLR receptors in our body, TLR1 to TLR10, and each of them has specific location and has specific rules in our body. For example, in some vaccine, scientists stimulate innate immune system and produce antibody stronger and enhance adaptive immune response in our body after vaccination. You know, in vaccination, use some substance as adjuvants for and these adjuvants enhance through enhancing innate immune system lead to boosting adaptive immune response and lead to adaptive immune response are activated stronger without these adjunct. So the ligand for these PAM recognition receptor can be used as adjunct for vaccination in different type of vaccine, DNA vaccine, RNA vaccine, protein vaccine, different type of vaccination. And what about PAM and DAM as a ligand for PAM recognition receptor? I told you PAM molecules as a ligand for PRR are made of proteins, nucleic acids, and also lipids, carbohydrates, and you know we have this structure in our body. We have a lot of proteins, we have a lot of nucleic acids, and why our immune system, our PAM recognition receptors in our innate immune system don't recognize our self structures, and only is able to recognize this structure on pathogens. What is the difference between PAM molecule on pathogens, and these structure, nucleic acid, proteins, lipid, carbohydrates in our body? They are different a little because, for example, the virus has double strand RNA, while we don't have double strand, we have single strand RNA, and also in some bacteria there are unmethylated cpG DNA, while we have some methylated DNA, and also in some microbes, specifically bacteria, proteins are initiated with N-formyl methionine, like pilin, flagellin in bacteria, and on the contrary, in our body, proteins aren't initiated with N-formyl methionine, but there are some lipids components in pathogens while they aren't present in our body. For example, some bacteria have negative bacteria, gram-negative bacteria has LPS as a lipid antigen, or has some gram-positive bacteria has lipopicric acid as a lipid antigen. Why we don't have this type of lipid in our body? And also about carbohydrate, some antigens like mononucleotide in fungi and also in some bacteria are made of oligosaccharide with terminal mannose residue, and in our body we don't have terminal mannose residue in our carbohydrate structure. So because of these difference between PAM and pathogens and these structures in our body, PAM recognition receptors in innate immune system can recognize, can differentiate between these self-structures from PAM's molecule and pathogen, and they only recognize PAM in pathogens, and they don't recognize our self-proteins or our self-lipid. And what about the features of PAM recognition receptors? In adaptive immune system, we have TCR and BCR. BCR means B cell receptor, TCR means T cell receptor. And they are specific a little diversity, and they are encoded by germline DNA. Why? On the contrary, in the adaptive immune system, BCR and TCR are generated through recombination, and we have a lot of type of TCR and BCR, and diversity of PCR and TCR is high in adaptive. But in innate immune system, we have a limited number of pan-recognition receptors, and all of them are able to recognize the pathogen as the same way. Okay, they aren't different between recognizing pan-recognition receptors for recognizing antigens. And so pan-recognition receptors have low specificity and diversity in our body. And what about specificity and diversity? You know, we have less than 100 different types of pan-recognition receptors in our body, and these number of limited number of pan-recognition receptors in innate immune system are able to recognize about only 1,000 molecule patterns. That this structure, molecular structures, are shared by different classes of pathogens. On the contrary, in our adaptive immune system, we have more than 10 to the power of 9, not 7, 10 to the power of 9 type of TCR and BCR on the surface of different pulmonary B cells and T cells. So these receptors, TCR, BCR, in adaptive immune system are able to recognize more than 10 to the power of 9 antigens, and they are able to recognize little difference between different pathogens specifically. And but I told you about pan-recognition receptor and how innate immune cells recognize antigens, pathogens, foreign substances through these receptors, and after that these cells are activated and lead to killing microbes through different mechanisms, through phagocytosis, degranulation, releasing ROS, reactive oxygen species, releasing nitric oxide, and the other things. And finally, they produce some molecules, some cytokines, inflammatory cytokines, and chemokines, and lead to recruitment of the other immune cells to help them to defend against pathogens. And but what is the phagocytosis? You know, phagocytosis.
Engulfing the foreign material.
It's the pathogen.
Yeah.
Okay. And after that,
It engulfs and binds the endosome or phagosome. Then
okay,
it is represented to the lysosome is digested.
it's correct. Exactly. When a pathogen enters to our body, our innate immune cells like macrophage, through their pan-recognition receptors, sense the pathogens and surround the pathogens and eat them, capture them, uptake them through endosome. And these pathogens enter to cytosol of phagocytic cells like phagosome and merge to lysosome and form phagolysosome, and after that, because of low pH, lysosome, lysosome and protease, lactoferrin, and the other degrading enzyme lead to processing, degradation, and killing the pathogens. And what about neutrophils as the first line of defense against pathogen? I told you the first line of defense is caused by neutrophil through phagocytosis, and I explain about phagocytosis. The other function of neutrophil is degranulation, means after sensing pathogens by chemical receptors. I told you some inactivating signals are received by these cells through signaling tail in the intracellular domain of high-recognition receptor, and these cells are activated and release all of their granule, and their granule contains some nitric oxides and some antimicrobial peptides and lead to killing the bacteria. And the other function of neutrophil that I mentioned in previous session, and I promised you to explain about it more, is netosis. Netosis is not necrosis. Netosis means neutrophil extracellular tract. The neutrophils, when aren't able to kill the pathogen through phagocytosis and degranulation, they sacrifice themselves. It's interesting. They sacrifice themselves for you, to protect you against different pathogens. And in this action, they release all of their components into extracellular traps, like their DNA, their histone, all of their components. And so they form a network in extracellular space, and in this network, the pathogens have been entered to our body, are trapped, and they are killed. So this means necrosis, neutrophil extracellular traps. And what about macrophage? Macrophage as another innate immune cells in our body is able to do phagocytosis, produce some inflammatory cytokines like interleukin-1, 6, TNF-alpha, and recruit the other immune cells to site of infection to help them for killing the pathogens, and also present antigen to lymphocyte T cell as antigen presenting cell. And on the contrary, we have another type of macrophage in our body. Is called alternative macrophage or non-classical macrophage. They are regulatory macrophage. After diluting pathogens, these alternative macrophage or monocyte start producing some cytokines like interleukin-10, TGF-beta. They are immunosuppressive cytokines, and they inhibit recruitment of immune cells to site of inflammation because they don't need the recruitment of immune cells. The pathogens have been deleted, okay? The war is finished, okay? They don't need that. So another role of this monocyte is producing some cytokines, some proteins, a little tissue repair. You know, after every war, in every place, not only criminals, not only politicians, but also all of the people who they play role in war, or who they don't play role in war. All of them are killed. All of them are damaged in every war. Okay, so after war, between our immune system and pathogens as target cells, not only the pathogens are killed, but some natural cells, some normal tissues are damaged. So these monocytes, through producing some molecules, lead to repair and remodeling of damaged tissue inside of infection, inside of injury. And what about eosinophils and mast cells and basophils as the other innate immune cells in our body? These immune cells, mast cells, eosinophils are very important. Yes, exactly, are very important in allergic reaction and also in defense against helminth parasites. And you are seeing in this slide defense against helminth parasites, and after producing interleukin 5 by T helper 2, these cells are recruited and these cells produce these cells are degranulated and release all of their granule and lead to delineation of these target pathogen. And but I told you in the allergic reactions, when allergen enter to our body, these allergens are recognized by mast cells dependent on IgE class of antibody and start degranulation and release a lot of mediators. The most common important mediators in mast cells granule are histamine, and histamine lead to inflammation and lead to allergic reaction. You know, these days they are after allergic reaction you usually use antihistamine and antihistamine because antihistamine neutralize the effect of histamine is produced during allergic reaction. And another innate immune cells is very, very important for presenting antigens to lymphocyte T cell, naive lymphocyte T cell, no effector lymphocyte T cells, are dendritic cells, and these cells are the bridge between innate and adaptive immune system. Another type of dendritic cells are called plasmacytoid dendritic cells. And these cells, like virus-infected cells, are able to produce interferon type 1 cytokines, alpha beta cytokines, and lead to inhibition of virus replication or lead to antiviral state in target cells. And NK cell or natural killer cells these days are using as cell therapy for cancers. You know, these days NK cell therapy is using for different type of cancers, for killing cancer cells, and these cells, in spite of lymphocyte T cells, I told you are restricted to presenting antigen by MHC, is not restricted to MHC molecule for recognizing the antigens, and they usually recognize the antigens and they are activate, and after receiving activating signals, and they start degranulation, release their granule like lymphocyte CTL, TCD8 positive cells in adaptive immune system. They are degranulated, release perforin granzyme, and make many pores in the membrane of target cells and lead to cytolysis of target cells. And usually target cells for natural killer cells are virus-infected cells and cancer cells. And also I told you NK cells as innate immune system is able to produce interferon gamma, and interferon gamma affect macrophage and enhance the phagocytosis and antimicrobial effect of macrophage. You know, all of our nucleated cells in our body, I told you express MHC1. MHC1 in normal condition act as inhibitory ligand for inhibitory receptor on the surface of NK cell. On the surface of every NK cell, we have a lot of inhibitory receptors and activator receptors. And if the activating signal is more than inhibiting signal, these NK cells are activated and are degranulated and lyse target cells. Okay. And why NK cells in normal condition are not able to lyse our normal cells, because all of our nucleated cells express MHC1 molecule on their surface, okay? And MHC1 molecule is as a passport or as a شناسنامه
Identity card.
identity card for our healthy cells, and show to NK cells, and they aren't killed by NK cells. They aren't lysed by NK cells. But in virus infected cells, you are seeing normal cells, normal hepatocyte. These cells has not been infected with virus and express MHC1. MHC1 bind to inhibitory receptor on the surface of NK cell and send inhibitory signal to NK cells. NK cell is inhibited. But when these target cells are infected with virus like hepatitis B virus, hepatitis C virus, these virus-infected cells miss expression of MHC1, okay, and they aren't able to express MHC1. So we can say they are missing self cells, okay, missing self cells. And on the contrary, they express a lot of stress antigen, stress proteins on their surface, and these stress proteins act as a ligand for activating receptors on the surface of NK cells. So, because the activatory signals are more than inhibitory signals, these cells are activated and start degranulation and lyse virus-infected cells. And the third line of defense in innate immune system is caused by some soluble proteins, and some of them like colectins, contraxin, they are able to bind to the surface of pathogens, and on the surface of our immune cells, we have some receptors for these contraxin, these colectins, contraxin like CRP. We have receptor for these molecules on our innate immune cells, and after binding these soluble protein in our serum to pathogens, they bind to the receptor on the surface of innate immune cells. These innate immune cells are activated and phagocytose them. In the other way, I can say these molecules act as opsonine. You know opsonine? Opsonine act as spices in our immune system and bind to the pathogens and foreign substances and make them delicious for innate immune cells to eat them. Okay? These
It is called opsonization.
opsonization. Yes, opsonization means the opsonins bind to their ligand on the surface of pathogen, make them delicious, and after that, the innate immune cells start phagocytosis them because they are delicious for them. Okay, and antibody is a kind of opsonine, contraxin, colectin, monosbinding lectin, CRP, all of them as an opsonine for our innate immune system. And another action is complement activation. Complement is mix of proteins, soluble proteins in our body. And you have a session about complement system, okay? We have a mix of proteins in our immune system, and every protein active another protein, and another active another, and they are activated in cascade manner, okay? And we have three distinct pattern or pathway of complement activation. Finally, in all of the pathways, three pathway of complement activation lead to making many pools on the surface of target cell, and complement activation lead to cell lysis. Okay, and these complement as soluble proteins in our serum lead to cytolysis, and also as a result of complement activation, a lot of pro-inflammatory cytokines are produced, and these cytokines, these chemokines, lead to recruitment of innate immune cells and lead to inflammation. Okay, I explained about three line of defense in innate immune system. The first line is barriers. The second line is immune cells, and the third line is soluble molecules, complement, contracting, fibroid. And now I wanna tell you about inflammation. Okay? I finished about 50 minutes. I finished this session. 50 minutes. 15, excuse me. No, 15. And what is the inflammation? Inflammation is very important for you, okay? Inflammation is non-specific and flowing way through which our innate immune system wants to control the spreading of the infection and deals with infection in our body. And in the normal condition, usually leukocytes are circulating in our blood circulation, and the number of leukocytes in tissues are low. Okay, and but when some pathogens enter to a specific site in our skin of finger, okay, or one needle enter to our to finger. So in this condition, these leukocytes are recruited or are extravasated to extra vascular site of tissue. And in this site, we have accumulation of leukocytes, plasma proteins, and also along them huge amount of serum exit from blood vessels and go into interstitial space, and so this event is called inflammation. But I want to explain about 3 steps of inflammation. 3 steps of inflammation comprehensively. In first session, I want to use everything I taught you, okay, in this part. I think this part of class, I know you are tired, okay, but this part of class is interesting, okay, and is useful for you. After entering pathogens into our body, or after damaging normal tissues, these dams and these PAMs are recognized by PAM recognition receptors on the surface of innate immune cell receptors like macrophages. These cells, because of receiving activatory signals, are activated and start producing a lot of different type of mediators like prostaglandins, leukotrienes, histamines, PAF or platelet activating factors, and inflammatory cytokines like interleukin-1, interleukin-6, and TNF-alpha. And in the step 2, cytokines are produced, are secreted, each mediators, each cytokines lead to some events. For example, nitric oxide as inflammatory mediators and cytotoxic mediators have some receptor on the surface of endothelial cells and affect endothelial cells and lead to contraction of endothelial cells, and in this way increase permeability of vessels, okay? Because these there are space in this condition, space between endothelial cells, and endothelial cell will be shorter than before, okay? So there are increased permeability, and also endothelial cells and also nitric oxides affect smooth muscles surrounding blood vessels, okay? After contraction of these smooth muscles surrounding blood vessels, these blood vessels lead to vasodilation, okay? And vasodilation means the blood volume is increased and blood flow is slowed down, okay? And so lead to redness because of increasing blood volume, and also heat inside of inflammation. And also I told you nitric oxide affect endothelial cells and lead to increasing permeability of the cells, and the condition is prepared for extravasation of leukocytes to extravascular site of tissue. So, as you are seeing, these mediators affect endothelial cells. For example, interleukin 1, interleukin 6, TNF affect endothelial cells and increase expression of some adhesion molecules. And these adhesion molecules bind to leukocytes and slow down their speed in blood circulation. So these leukocytes, after binding to these adhesion molecules on the surface of these endothelial cells, bind to them, and because increased permeability of blood vessels, extravasate to extracellular site or the site of infection, the pathogens enter there. And so these cytokines, through the action I told you, phagocytosis and netosis, opsonization, complement activation, kill the pathogen in this site. So I told you another factor is produced is platelet activating factor, or PAP. PAP leads to platelet aggregation and is a clot, and in this way lead to inhibition of bleeding in our body in site of injury, okay? So another aim of inflammation, apart from defense against pathogens and killing pathogen, is repairing, okay? So in this way, inhibit bleeding and form fibrin around platelet aggregation, and so finally these cells I told you through different mechanisms kill the pathogens. And but in the step 3 of inflammation, after accumulation of many leukocyte into site of inflammation, into site of infection or injury, the third outcome of inflammation is repairing and tissue remodeling, because some leukocyte like macrophage, like macrophage, like site of infection or site of injury, and also alternative monocyte. I told you play in regulation and modulation of innate immune system, start producing anti-inflammatory cytokine and stop the recruitment of immune cells, and also start repairing. So you are seeing typical signs of inflammation: heat, redness, edema, and swelling. Okay, edema or swelling. Okay, and so also if the inflammation is severe, people usually experience pain because of some mediators like prostaglandin, icosanoid, and something like this are produced by immune cells in site of inflammation after recruitment to site of infection. After affecting nitric oxide on the smooth muscle surrounding blood vessels, I told you volume of blood is increased, okay, and also these increased volume blood lead to redness and heat. And also I told you we have swelling or edema because of recruitment and accumulation of a lot of immune cells inside of infection or inflammation, okay? And also because of entering a huge amount of serum or blood fluid along with leukocyte to site of infection, we have swelling or edema. And so inflammatory response can be effective. Why? Because protect us against pathogen, because control spreading the pathogen until the innate adaptive immune system starts removing the pathogen. And also, inflammatory response initiate tissue repairing after removing the pathogens, starts producing some mediators and lead to repair and tissue remodeling. And what about inflammatory cytokines? You know, inflammatory cytokines in innate immune system, I told you, are interleukin 1, interleukin 6, and TNF-alpha. When some pathogens enter to our body, our innate immune cells, after recognition them by PAM recognition receptors, are activated and produce these cytokines. And these cytokines affect endothelial cells and increase expression of some antigen molecules on these cells, endothelial cells, and lead to recruitment of a lot of innate immune cells into site of infection and lead to inflammation. Okay, is good because protect us against pathogen and control spreading the pathogen, and after that repairing the normal tissue, okay, that have been damaged during war between immune system and pathogen. And but this is a local effect of inflammatory, and but there are some systemic effect of inflammatory cytokines that they are effective for us. Also, they are systemic. So sometimes these cytokines are entered through blood circulation, okay, and they affect our brain and lead to fever. And fever, you know, fever is high temperature, and high temperature inhibit growth of microbes and is beneficial. And also affect liver and produce acute, some acute inflammation proteins like CRP or C-reactive proteins, And you know, CRP act as oxalate in our body and finally lead to oxidation and phagocytosis. And during inflammation, when the cytokines enter to our blood, it can affect our bone marrow. It's a site of producing a lot of immune cells in our body, leukocytes in our body. Affect bone marrow and tell bone marrow, please produce more leukocyte for me. Please produce more leukocyte because I need more leukocyte, more soldiers, leukocyte as soldiers to deal with pathogens. Okay, so all of them are beneficial for us. Local effect and systemic effect of inflammatory cytokines, but sometimes inflammatory cytokines have pathologic effects. I told you in the first session about cytokines. Some inflammatory cytokines are associated with some autoimmune disease, inflammatory disease, some cancers. And you know, when TNF is produced in a huge amount in our body, these cytokines affect cardiac cells and reduce contraction of cardiac cells and lead to low output in our heart, and so lead to low blood pressure and lead to septic shock in our body, okay, because of low blood pressure. And also TNF affect endothelial cells, and after affecting endothelial cells lead to induce production of tissue factor. Tissue factor is very important for population. Yes, lead to thrombosis, and also affect muscle cells, skeletal muscles, and lead to insulin resistance and abnormality in metabolism of these cells and weak contraction of muscles. So when these inflammatory cytokines like TNF are produced in a huge amount, lead to shock in our body. Okay, so two different mechanisms I explained to you. So in this session, as third session for today, I'll explain about innate immunity in third line, and finally I told you how inflammatory responses are produced in 3 steps and detail, and I told you symptoms of inflammatory reaction or responses in our body, and about beneficial and protective effect of inflammatory cytokine. If you have a question, I'm ready to answer your question. If not, I show you quiz. Okay, the quiz. The quiz is really easy. Which one is correct? Inflammation can be protective or harmful.